It's an honor to be here, and I'm excited to tell you about our work trying to understand the remarkable protein machines that drive cell division. So the goal of cell division, at least from the cell's perspective, is to make an exact copy of yourself. I would argue the most important thing a cell needs to copy accurately is its genetic information, which is carried on chromosomes in this video shown in blue. So accurate chromosome segregation is essential for the maintenance of cellular and organismal fitness. For this reason, it's not surprising that errors in this process are associated with numerous human diseases, including all of the trisomies, including Down syndrome, as well as cancer. What blows my mind is the sheer number of times that this has to occur properly. So it is estimated that over an average human lifetime, 10 quadrillion cell divisions need to occur. My lab is interested in understanding the protein machines that drive this process and allow it to occur with such high fidelity. Before I tell you a little more about our science, I just wanted to briefly tell you about my scientific path. So I grew up next door in Colorado. I decided to move slightly east for undergrad, going to Carleton. This is really where my passion for science started, majoring in biology. I went on to work at a small biotech company called NatureWorks, and this company has and had the honorable goal of making biodegradable plastic out of corn. I was lucky enough to work with some really bright people at this company. We made some important discoveries that made commercialization of this product possible. This is also where I realized my passion for understanding our world, and really for using this basic knowledge for the betterment of mankind. And in this case, reducing our carbon footprint. And it really was, this is really what drove me to want to continue using basic research as my life's work. Okay, so I decided to go to grad school, going to MIT, working with a fantastic scientist, Dr. Angelika Amon. I'll say more about her in a few slides. I continued my training working with an equally fantastic scientist and mentor, Sue Biggins at the Fred Hutch. Then I was lucky enough to start here as an assistant professor in 2019. So I view moving to Utah really as closing the circle. Of course, bringing me back to the Mountain West and the wonderful mountains we have here, but really also the amazing community we have and filled with amazing colleagues and scientists. And so I really feel honored to be here. Okay, so back to our science. We are interested again in cell division and the process of chromosome segregation, which is a highly dynamic process. So in this movie, what you're going to see is the cell's chromosomes are marked in white. The cell's mitotic spindle, which is the segregation machinery, is labeled in green. And what you can see is that the chromosomes are all going to attach to this mitotic spindle, get pushed and pulled around in the cell, ultimately aligning all of the chromosomes in the middle of the cell before equally partitioning them to opposite poles. So if we zoom in at what is actually carrying this process out, the mitotic spindle is made up of these dynamic protein polymers called microtubules, and it's actually their assembly and disassembly that drives chromosome segregation. The way that chromosomes attach to microtubules is through this highly conserved protein complex, the kinetochore. And it's really this kinetochore-microtubule interface that my lab is interested in understanding. Okay, so I mentioned chromosome segregation is really important. It's essential for cell viability. What's surprising is how error-prone this process becomes in particular disease contexts, especially in cancer. So to highlight this point, I'm going to show you something we call a karyotype. So a karyotype is where we can basically look at all of the chromosomes that are in a cell, line them up so we can count the number and their composition. So this is what our karyotype of a normal cell should look like. Almost all of the cells in our body look like this. We have 23 pairs of chromosomes, one from mom and one from dad. If we look at a karyotype from a cancer cell, here on the right, you can see that it looks very different. Hopefully you can appreciate immediately that not all of the chromosomes are carried in 2 copies, and in fact, many are carried in much more than 2 copies. This state of chromosomal imbalance is known as aneuploidy, which is a hallmark of cancer. So greater than 95% of solid tumors are aneuploid. And again, this is very detrimental to the cell, so this is a bit surprising. But it's been shown that this alone can contribute to tumor progression and perhaps even tumor initiation. So again, my lab wants to understand how this process occurs in normal cells, such that we can understand how it becomes defective in human disease. So one of the ways that this can become defective is if the cell is not able to sense and correct erroneous attachments. So let me tell you how that works. So in order to segregate your chromosomes accurately, you need to make attachments that look like this, attachments to microtubules that emanate from opposite poles. However, when attachments are initially made, the majority of attachments are made incorrectly, sort of schematized in these top two examples. So the cell needs to have a way to detect and to correct these erroneous attachments. And the way the cell does this, I still think is super cool, and that is through tension. So incorrect attachments generate a low amount of tension. The cell can recognize this and can trigger the kinetochore to release these attachments, giving the cell another chance to make a correct attachment. Conversely, these correct attachments generate a high amount of tension, and these are selectively stabilized. So really the question we're interested in is, how does tension alter these attachments? So this question has proven incredibly challenging to address in cells because these attachments are highly dynamic, and measuring and perturbing these physical forces has proven challenging, if not impossible. So our approach has been to try to recreate these kinetochore-microtubule attachments in the lab. Okay, so in order to do this, we need to be able to pull these protein machines out of the cell. And so we've developed methods that allow us to do this, purifying both the kinetochore as well as microtubules from cells. This is what one of these attachments looks like in the lab under a very powerful microscope. We can then use a different microscope, but equally powerful, to apply user-defined forces across this interface in the lab. And so using this approach, we've made a couple of interesting and I hope important discoveries. One of them is that one of the ways the kinetochore actually responds to tension is that it functions like a children's finger trap toy. Basically, the stronger you pull on it, the harder it holds on. And then we've gone on to identify some of the first factors that are involved in this pathway. So with these observations, we've been able to secure funding from a number of sources, including AMIRA from NIH, funding from the Damon Runyon Cancer Research Foundation, the Pew Charitable Trust, as well as Fight for the Fight. And we're really excited to use this funding to go on and try to understand, in addition to other questions, how this molecular finger trap actually works. Okay, so I just want to end by coming back to some lessons I've learned, and really the foundation that was set for me by some really amazing mentors. So Angelica and Sue. So Angelica unfortunately passed away a few years ago prematurely, but the lessons that I learned from her and Sue really have lived on, and I hope I will be able to pass on to my current trainees. So those lessons are to be supportive and to find a place where you can be supported, to be fearless and creative in your science, and related to that, to ask the tough questions. Don't always take the path of least resistance. That often just leads you to the least interesting observation. To be your own harshest critic. Don't let a reviewer do that for you. And finally, I think most importantly, to follow your curiosity and really to have fun. And so I feel very fortunate to be in such a supportive place here at the University of Utah, where I'm happy to try to take these lessons I've learned and pass them on to my current and future trainees. And I just want to end by thanking them, as well as our collaborators and this amazing community we have here. It's really all of these combined things that make this job really fun. So thank you.